Okay. So, the system that I am talking about is really important in the context that I am going to provide first. So, you could have species organized in this adaptive landscape, and this is one way we think of speciation. There could be multiple ways. You have natural selection, which is driving populations apart, and you have this. Are you all familiar with adaptive landscape? No? Okay. So, adaptive landscape is this idea which helps you visualize what's going on in terms of phenotype and combinations of phenotypes, and what that does to fitness of an organism and what that will do to adaptation. So, this is the basic model. So, you have phenotypes here, and we are just showing two. You could have an n dimensional phenotype space. For simplicity, we just use two phenotypes, and then those combinations of phenotypes, depending on what those phenotypes are, may make some areas fitness peaks. So, this is fitness. So, greater the height, better the fitness of that phenotypic combination, and you have some areas where fitness is really low. For example, you can imagine there is flower color and let us say nectar. So, if you have a certain combination of flower color and nectar, that is going to attract certain kinds of pollinators. If you have another flower color and another kind of nectar, you are going to attract another set of flowers. So, you can imagine that combinations which go together of those two very different kinds of phenotypes might provide the plant greater fitness because it attracts right kind of pollinators. Whereas, if it has one type of color and another type of nectar, then that does not attract either the nectarivores completely or nectarivores of one kind or another. And if that flower color is the cue that different communities use, then you are attracting wrong kind of pollinator for the wrong kind of nectar, right. So, then that will become a fitness valley. So, that is how you can imagine any set of characters, any set of phenotypes making the landscape rugged. It is like a mountainous landscape rather than a flat landscape. So, you could have these empty niches, meaning certain fitness peaks which are not occupied by any species. So, if you land upon that fitness peak, or at least in that neighborhood, then you can climb that fitness peak, and then you will have greater fitness, and then you have that, you have occupied that niche. So, fitness optima species do better there, and there are a lot of complications to this. I will not go into the details. For example, this fitness landscape is not constant, it is changing, it is a completely dynamic landscape. So, anyway, that's complication you don't need to worry about right now. So, you can think of species and speciation as a process which is happening in this particular fitness landscape. And you could have fitness, I mean you could have selection of various kinds. You could have natural selection, you could have sexual selection, where now these phenotypes will be related to something to do with mate attraction or other things related to sexual selection. You could also have polyploidization and hybrid speciation. So, polyploidization, for example, with the same traits, but just with two doses of something might increase your productivity, for example, which might give you a fitness advantage, and so on and so forth. So, you could have various ways in which speciation happens, and you could have different ways in which hybridization can. So, here you do not necessarily need to worry about it. This is where it becomes interesting. So, polyploidization, as you know, is a very common process. It is supposed to be widespread among plants, not so much among animals, and the reason for that is that it is considered that, or at least until 10-15 years ago, it was considered that hybrid speciation was very unlikely in animals, mainly because there are lots of barriers which plants often do not face, or not as much. So, first, hybrids are often sterile, and the way this is not the reason why this is not a big problem for plants is that a lot of them can reproduce asexually. So, if it is a hybrid individual which cannot produce gametes successfully or attract right kind of pollinators and so on, if it is a grass species, for example, it can just start putting runners and spread. It doesn't need that one individual doesn't die necessarily. There are various ways in which they can reproduce. First hybrids may be weakly reproductively isolated, so they backcross and therefore merge into the ancestral species, into the original species, and therefore they may not actually evolve separately. Plants, for example, avoid that problem by if it's, let's say, polyploid, then its chromosome number is going to change, and then it will be, because of the way gametes are formed, will become incompatible with either parental species, right? This is something you have done in genetics. So there are all these different problems that animals face, plants don't necessarily face, because of which you have lots and lots of cases of polyploidization and hybridization or hybrid speciation in plants, not so much in animals. And the way we think about this is that the only way you could have hybrid speciation is if your parental species are sitting on two different fitness peaks. You may have hybrids which land in a fitness valley, and therefore they don't have any evolutionary future. Now, just by chance, it may so happen that the hybrids may land on an unoccupied fitness peak, in which case they can take off as an independent population, but the chances of that happening are really, really low. Now, if they occur in an area where they are in a fitness valley, but either because selection is relaxed or some other reasons, maybe they can climb up on a nearby fitness peak, and they could potentially have an evolutionary future at least after a few generations. So, you could have a chance occurrence, or because of relaxed selection, some movement in this landscape which might make hybrids successful. But again, the chances of any of that happening are generally low, especially in animals. So, that is something that had been the understanding for a long time. Of course, a few hybrids between species were known which have given rise to daughter species. As I said, this is really common in plants. We have wheat and many other common plant varieties that we see every day that have hybridization at various levels in their evolutionary history. So, really, really common. According to some estimates, some 40 to 60% of all plants may have polyploidization, and by necessity hybridization, hybrid speciation in their ancestry. So, quite an interesting phenomenon there. But for animals it was considered rare until recently, and some people still say that, you know, very good cases of hybrid speciation are rare. And this is where I got interested in this phenomenon, mainly because when I was a graduate student way back in 200s, early 200s, somebody described a new species of what is called a tiger swallowtail from North America. So, this dark green area is where it occurs, Papilio appalachiensis, Appalachian tiger swallowtail. They are called tiger swallowtails because they have this black stripe yellow pattern like tigers. So, these are big butterflies, these are massive. And what made big news is that this area has been occupied by Europeans for at least more than 250 years, and when I say occupied, really well occupied. Things have been collected from there for a long time. These butterflies and all populations had been known for a long time, and in spite of that, it was not until 2002 when this population was identified as distinct and named, and the two people who named it called it a new species, and they even speculated about where that species came from. But before I go there, I will just mention to you that this had an ecological context, and on the previous slide I had said, you know, this 40-year old, or rather 40 years of studying this particular group has provided us a lot of insights into how evolution proceeds. So, we had a lot of information about this. So, the key components of this I will just mention quickly. So, what was known in this area is that you have one species, the eastern tiger swallowtail, Papilio glaucus, which is its range here, and then you have the Canadian tiger swallowtail here, which is its range here in blue. And this dotted line is Batis philonor. It is a toxic butterfly, and in this species, eastern tiger swallowtail, male looks like that. There is a male like female form, and then there is a mimetic female form which mimics this. Okay, and in the Canadian tiger swallowtail you don't have that black form, you only have male like female. Okay, and this area here is the hybrid zone between these two populations. So, for a long time Canadensis was considered a subspecies of glaucus, and in the 1991-92 our collaborator Mark Schreiber said that there are enough differences between these two that we should really call them different species. Now, this is still an unsettled matter, because if you look across these two species, their genome is exactly the same, there is no differentiation between them. The only differentiation you see is with a few key genes and few key traits. So, whether you want to call it subspecies or species does not matter, but the divergence is quite interesting in terms of its evolutionary significance. So, Pavulan and Wright, these were the two guys, and in 2004 they wrote another paper describing new distributions and phenotypes and so on. So, this is what they showed, and Mark Schreiber has been studying this system for 40 years. He just retired recently, but he knows the most about this system. So, this is what happens. You have glaucus here, Appalachiensis here, this presumed hybrid species which is endemic to Appalachian mountains, and then you have Canadensis here. So, remember this distribution, Appalachiensis is only in Appalachian mountains which run like this, okay. And then you have the cold adapted species here and the mimetic species here. So, this is what you see. In glaucus, it is adapted to a warmer thermal habitat. It has a pupal diapause which is facultative. So, lower down in Texas and Florida, lower states in the United States, because it is warmer for the large part of the year, they have several generations per year, and up north near the Canadian border, for example, they have a single generation. You cannot have more than one generation in that area because it is just too cold most of the time. So, the pupal diapause here is facultative, here it is obligatory. Voltinism has at least 2 generations per year, this has only a single generation per year. Survival on this one particular plant is low, it is high here, and then you have several other features related to mimicry and dimorphism. So, sexual dimorphism is present in glaucus, you have black form and male form, and then one female like form. Here you do not have sexual dimorphism, it is monomorphic. There is Betzian mimicry, and that mimicry itself is female limited to one form. And then on another plant there is high survival here, low survival there. So, Pavulan and Wright looked at this Appalachian population and said, oh look, you have this warm adapted species and you have this cold adapted species, and then you have a population in this area. Now, remember I said Appalachian mountains, these are cold mountains, even when they are in the south because of elevation they are cold. So, in some sense they are like greater latitude. So, they said no matter where you are in the Appalachian mountains, these things, the Appalachians is the new species that they described is like Canadensis for diapause related traits, and it is like glaucus for mimicry related traits, because the distribution of Philanor, Batis philanor, the toxic butterfly, limits how far north the mimetic type can go. Okay. So, they said for mimicry related things, Appalachiensis looks like glaucus, for cold tolerance related phenotypes it looks like Canadensis. So, it must be a hybrid species, and remember this is 2002-2004 when people were just beginning to come up with Nature and Science papers, so showing that here was one hybrid species, there was another hybrid species. So, this topic was really hot at that time. So, I said okay, this sounds interesting. As a PhD student you are always looking for interesting things to do, right, and that continues into your career. So, another interesting thing about this is that all the cold tolerance genes here are Z-linked. Remember in butterflies and birds, females are heterogametic, they have 2 types of chromosomes and males have a single type of chromosome, and to distinguish that from XY system we call them ZW. So W is found only in females, Z is found in males and females. So the cold tolerance genes are Z linked. All these, this is again Mark Scriber's work. He had not identified those genes, but he had mapped them by doing interspecific crosses and found out what are the differences in that. And he knew that these were Z linked based on that work. And mimicry in this species is W linked. So you can take babies of this particular female. All the females will always be like, all her daughters will always be like that, no matter how many times you breed them. And daughters of this female will always be black. So there is nothing that is shared between the autosomes. It's a completely W-linked trait. So people have looked at this for some time and have done some sort of experiments to suggest that it's not mitochondrial factors, it is W-linked. Okay, so or at least that is the best explanation given the data. So this was the system we started to look at because this appeared to be a hybrid species. So the first thing we did was we took a bunch of traits. These are called AFLPs. These are traits meaning molecular traits. So these are markers that are called AFLPs. These are restriction site based markers. Don't worry about the kind of markers these are. What you do see is that when you look at all the species within tiger swallowtail groups, you have these out groups. So this is just outside the tiger swallowtails, and then you have these 8 different taxa under 8 taxa under tiger swallowtails, and we showed that glaucus forms one group, canadensis has another group, and this is somewhere in between. Okay, this is using that. So what appears that if this is a distinct species, a hybrid species, it must have got these mimicry genes and that trait from glaucus, and cold tolerance gene from appalachiensis, which is why it comes in between. This is the crudest approach you can have. So we decided to go a step further and look at specific integration. So you are familiar with integration now. So is there any specific pattern to integration? This is what we looked at. So here each column is a particular individual and each row is a particular locus. So there are so many SNPs in CO1 gene, so many SNPs in single nucleotide polymorphism in these Z-linked genes, and each one of these positions is numbered. So you won't see the details here, but these black scribbling are basically base number. So this is SNP for each individual. That is what you see here. And what you see here is, another thing, the reason why we use mitochondrial gene here rather than W-linked markers is that we don't have any W-linked markers. People have struggled for decades to find W-linked markers which work in any kind of system, and we just don't have anything. There are some in bombyx, the silkworm, which work only in bombyx. But because W chromosome is found only in females, and that is passed down to only daughters, and mitochondria are also passed down only through the daughters. Basically, there is a linkage between mitochondrial genome and W chromosome, right? So you could use mitochondrial genes to track evolution history of W chromosome. So even if you didn't have W chromosome, there's an easy way out. So that mitochondrial gene actually shows history of the W chromosome as well. So this is what you see. Glaucus, here purple means a glaucus-like haplotype, and pale blue means a canadensis like haplotype. So for all the genes, you have a solid canadensis haplotype, and for all the genes, you have a solid glaucus-like phenotype. But in 2 genes, there is a lot of integration from canadensis into this, not so much from glaucus into canadensis. Okay, are you following? This is getting into genetics and population genetics. So if you don't understand, stop me anytime. And then when you look at appalachiensis W chromosomes are like glaucus, right? It's the exact same haplotype. And when you look at the rest of the genes, it's a mosaic of glaucus like haplotypes and canadensis like haplotypes. You see a lot of what is called admixture a signature of hybridization. Okay. Now, another interesting thing to see is that the reason why we have chosen all these Z-linked genes is that these are related to cold tolerance and melanin production, which are involved in in this ecological selection between which is separating glaucus from canadensis, and that is you have, oh, I didn't tell you the entire story. There is one Z-linked marker which is influencing mimicry to some extent. That is not very well understood, but there is something there. So, all these things are linked to, most of them, to cold tolerance. So, for canadensis you do expect that most of it has come from, for Appalachiensis, most of these Z-linked markers would have come from canadensis, because these are much more tightly linked to cold tolerance. So, indeed you see there is much more blue here for Appalachiensis than there is purple. Okay. So, you have very selective admixture based on the genetic basis of these two suites of traits. Mimicry is one, for which Appalachiensis looks like glaucus, because it shares that mimetic phenotype with glaucus, and then for cold tolerance you have Appalachiensis looking more like canadensis. So, it seems like it has selectively inherited these two sets of characters from the two parental species. So, we went a little bit further and said there is a program called structure which can form clusters based on your genetic data. So, we said if you give cluster all the information we have and ask it to make number of populations by clusters, how many populations will it make, where will these individuals go? That is what is being shown here. So, again, each column here, this one here, is an individual, and this is the probability that a contribution, genetic contribution in that individual has come from this or that. So, again we have used the same colors. So, what you see is that all the glaucus individuals are more or less like solid glaucus haplotype, and this is now at the genome level. So, the previous slide was just looking at mitochondrial gene and 6 Z-linked genes. This is using AFLPs, that restriction based marker. So, this is genome level data. So, across the genome, once again you have a pretty solid canadensis block and a pretty solid glaucus block, meaning that, oh, and here K means number of populations that you predefine. So, you put in all the individuals, all the molecular variation that you have in your data set, and tell structure there are only 2 species here. So, how will you put all this variation into 2 groups? So, it identified one group as a solid canadensis, so all the individuals showed cohesiveness for all those markers, and all the glaucus individuals showed cohesiveness for that, and this is what is interesting. So, this is Appalachiensis, our putative hybrid species, and these are lab generated F1 and F2 hybrids. Okay. These are cool things that you can do with butterflies, which you can't do with every other thing. So, Mark Schreiber had generated lab hybrids by putting canadensis and glaucus together, did it for 2 generations, and that is what's gone here. So, Appalachiensis and lab hybrids look very similar. They are about 50-50 glaucus and canadensis. So, at the genome level, Appalachiensis is an admixed population between canadensis and glaucus. Now, we said to structure, we told it to put everything in 3 clusters. We said instead of 2 species, now we tell you there are 3 species. How would you cluster these? So, what cluster did was it still still identified one glaucus cluster, another canadensis cluster, and then it put Appalachiensis as the 3rd cluster, meaning that it was cohesive, and it still put lab generated hybrids as 50-50 canadensis and glaucus. Okay. So, there is genetic variation such that Appalachiensis has a few unique alleles, which are different from lab generated hybrids, meaning what is there in glaucus and canadensis. And this is what's shown here. If you look at all the AFLP markers, there are that many which are glaucus like, there are that many which are canadensis like, there are that many which are intermediate between those two, and there were 16 unique markers which are found in Appalachiensis and not in the parental species. Okay. So, that is what you see here. And if we asked structure to make 4 populations, it started to pull apart different subpopulations within glaucus rather than make this separate or subdivide this. So, anything beyond 4 populations, you are just splitting, you are just identifying population structure rather than trying to identify what are higher level entities like species. So, we just stopped there, and the score for this one was the best. There is a particular statistical test I won't go into the details of, but that showed that this was really the best way to represent all the genetic variation. So, that was really interesting. Traditionally, what people had done was you do some sequencing and you say this particular putative hybrid species has some markers like that species, other markers like another species. The reason why this was a unique study was that we first showed there are ecologically important traits like cold tolerance and like mimicry, which are differentiating between which are diverging glaucus from canadensis, and that this putative hybrid species has an interesting mix of those two. In Appalachian mountains it needs cold tolerance, but because there is opportunity for mimicry, you have that Z linked, I mean W linked trait there as well, which is ecologically useful. So, it had that interesting mix of interesting mix of traits, and that you saw genetic evidence for that specific admixture. One set of characters coming from one species, another set of characters coming from another species. The next step that we were able to show was that at the genome level, this hybrid population is distinct, is admixed between the two parental species, but it is distinct from lab generated hybrids, meaning that there is something different in it than recently generated hybrids. So, to demonstrate that this was really an ancient hybridization event, we again looked at the AFLP data, and this is an estimated divergence time in years, and this is posterior probability of when divergence would have happened. So, again you look at all the variation in the 3 groups, Papilio-glaucus, canadensis and Appalachiensis, and based on that variation you try to estimate how far apart species pair wise divergence occurred. So, divergence between glaucus and canadensis, divergence between canadensis and Appalachiensis, divergence between Appalachiensis and glaucus. Okay. So, you have 3 divergences, and this is what you see. This is divergence between canadensis and glaucus, the 2 populations separated by thermal gradient in North America, and that divergence is about 600,000 years ago, about 6 lakhs years ago. And if you look at divergence between Appalachiensis and glaucus, Appalachiensis and canadensis, it is about 100,000 years ago. It is actually about 90,000 years ago. So, what is interesting here is that the parental species diverged a long time ago, and what we think is a hybrid species diverged from both the parental, what a putative parental species around the same time. Okay. And that is not such an old occurrence, but it is still old enough that it is not exactly like these F1 and F2 hybrids that we had generated. Okay. So, this is much more ancient compared to current ongoing hybridization between glaucus and canadensis, but because of this peculiar geography of Appalachian mountains, somehow this had happened. So, the idea that we have is that I just mentioned this divergence, and that is a classic signature of hybrid speciation. The parental species diverge much earlier, and the new hybrid species diverges from both the parents at the same time. If there is a bifurcating regular phylogenetic diversification, evolutionary divergence between populations, you will expect that there will be some staggering between when these populations diverge from each other. Now, it is possible that glaucus diverged from one species much earlier and then the second species in relative terms, but they were still somewhat closer in time compared to that 600,000 years of divergence that I showed between glaucus and Appalachiensis. But it's somewhat unlikely, especially given all the evidence that when you look at the genome, it is a mix of both the species. That you won't get if it was not a hybrid species. So when you put all of this together, that was one of the best examples of hybrid speciation at that time, and this was 2011. This is still one of the best examples of hybrid speciation. Now, as I said, some people might say, oh, but these are not really hybrid, these are not really different species. We don't care. The point is that there were two diverged populations based on some ecological selection, and that you have a new population which has evolved as a result of intermixing between the two ancestral populations which have been locally adapted to cold and to mimicry. That is the point. Taxonomists can go on arguing about whether this is 600,000 years of divergence is enough, or when rest of the genome is similar, it's only few critical genes which are pulling these apart, and so on and so forth. But as I said, population to species continuum, remember that. What is important is the evolutionary process underlying rather than the names that you give it. So this is what I think happened. So those two estimated divergence dates that I mentioned, 600,000 years ago and 90,000 years ago, are interesting because at both times there was a slightly warm period. So Earth's climate goes through glaciation and interglacial periods, right? So both 600,000 and 90,000 years was interglacial period. So it's possible that because thermal biology, thermal landscape plays such a big role in these species, something is tied in their biology with these climatic cycles. So and again, exact dates don't matter so much. What we think happened was that the boundary, the hybrid zone between these two species probably kept on going north and south depending on how cold it is, and that has probably been happening all the time, even across different years. If there is cold or warm climate, then you expect that to move at least by several miles here and there, right? So around 90,000 years ago, what might have happened was that probably canadensis was in the Appalachian Mountains all the way down because it was cold there. Even if it was warm right a few kilometers east or west, because in the plains, in the mountains it was colder, so probably canadensis was found all the way to the south in the Appalachian Mountains, and because of interglacial period, climate was warming, and that was pushing this entire relative ranges north and the hybrid zone north as a result. So with warming, probably glaucus started to invade the mountain a little bit and north a little bit. Thermal climate is the same there, right? Thermal zones. So as it was invading up in the hills, probably it came in contact with the canadensis like populations in the mountains, and there was very brief quick hybridization event, which is why you don't see much staggering in how it diverged. If it had been much prolonged bout of divergence, much prolonged bout of hybridization, and as I mentioned, this hybrid zone still exists. You still find hybrids in this area between those two. So if this had been ongoing, then you won't see 90,000 years estimate. You would see this as ongoing hybridization, right? You don't see that. And given that Appalachiensis has got the black female form from glaucus, it was probably that males of canadensis were mating with females of glaucus, which caused that hybrid population to arise. If it had not been that, then you would not get that black form and the yellow form like this. You would get things like this because there is no mimicry then. if W did not come from glaucus. And what is interesting is that Schreiber had shown that glaucus males, these males, care what female they mate with. So if you give them a choice between this female and that female, they preferentially mate with female of their own species. If you give them canadensis a choice between this female and that female, they don't care. You give them any female which looks like that, they'll be happy with it. So you can imagine what has happened about 90,000 years ago, because, and probably this preference is old as well. Maybe these males did not like to mate with these females when they were invading here, whereas these males didn't care. So the Appalachian hybrid that you see here is a result of all the Z-linked markers, cold tolerance genes that came from males of canadensis, and all the mimetic alleles that came from glaucus, which is why you have this very interesting mix of cold tolerance and mimicry in the hybrid population. And that was driven by both retreat of canadensis northward and upward in the mountains because of climate change, and because Batis probably moved up. So Batis is another interesting thing. If you look at the butterfly can tolerate cold, no problem at all. If you look at its host plant can't really grow at cold temperatures. So the range of this butterfly is limited by its larval host plant, and therefore the range of this female form is limited by the larval host plant of this butterfly, right? So as this butterfly was invading northward, that female form, that W allele, also moved up, and moved up in the mountains as well as up north, and that is what gave rise to Appalachiensis. That seems to be the story that appears based on all the genetic evidence that we generated. So because you have this interesting ecology and very strong genetic evidence from markers which are tied to those specific ecological traits, I think it is still a very nice demonstration of how you could have things which are divergent, these two, based on the ecological factors which drive life history traits of these butterflies, and how you could have hybridization, introgression leading to distinct populations which draw initially from the two parental populations and make an interesting mix which helps them occupy this new niche now. So again, going back to the adaptive landscape, this is sitting on a cold adapted peak, this is sitting on a warm adapted mimetic peak, and you have this interesting case here where climate change and movement of this particular butterfly has created a new peak, which is cold tolerance from one species and mimicry from another, which this hybrid was able to occupy. So that's the story. So I will, I think I had few slides, but I could stop soon. I'll just make this point very briefly. Now the reason why we were able to do that hybrid speciation study was that Mark Schreiber had been studying that system for 40 years before we cracked this, and we really do not have many people here which have done that. The only two people I can remember right now are Raghavendra Gadakkar and Raman Sukumar, both at CES, who have been looking at a system, Rhopalidia, wasps, and elephants and forest dynamic in the other case. But apart from them, there are a few others who have looked at monkeys, for example, for a long time. But Raghavendra Gadakkar has been particularly outstanding in the sense that he has focused on this one system and repeatedly kept up with what you can address with this system in what evolutionary biologists or behavioral biologists are interested in, and that is the kind of models that would be very interesting to look at in the Indian context. And if you have more and more of those, and maybe some of you will become that. In fact, if you don't know what to do with your scientific career, maybe take some inspiration from this. Identify whatever fascinates you. In my case, it was butterflies, and especially this Papiliopolitis butterfly. So as I mentioned, in my lab what we like to do is look at a particular system. In our case, it's sexual evolution of sexual dimorphism, evolution of population, I mean not population, evolution of polymorphism and all the ecological dynamics that follow uh, or influence evolution of sexual dimorphism and mimicry and so on. So we have been trying to do that. So just find out what is it that really drives you, what is it that really excites you, and if you stick to that, think a lot, look around a lot, you are going to find something which is very exciting. And I won't go into the details of this. If you really want to see the peak of this kind of work, look up Peter and Rosemary Grant. They have worked on Darwin's finches for more than 40 years now. Amazing piece of work, has informed so many ideas in evolutionary biology and ecology. That is something that we can do better because we just have much more biological material here than what a lot of European and American biologists have, right? So if you just pick up things that you really like around, you are going to see something, you're going to find out something really interesting, you're going to discover something absolutely new that is going to stun everybody. And I'm not talking about that work here today, but if you want to look up what we have discovered in Papiliopolitus in terms of what determines its wing patterns, that is a butterfly that you see in your backyard, and we found something very cool about it that everybody is excited about. And we are continuing that with other papilios. Find out what really you like, and if you persist, I think you're going to have a great career. Okay, this is something I talked about earlier, so I won't dwell on this earlier, but again, if you do find one inviolate law in biology, let me know. And if you don't, think about why you don't see it and what to do about it. You've probably heard of this term, physics envy, right? Have you heard that? Yeah. And my response to that is, we study something way more complicated and way more interesting for that reason. So we don't have to have one thing which applies everywhere, which is what makes biology more interesting to me. So they think you need some laws. If you don't have laws, then it's not science. You know, everything is determined by what you're looking at. I don't have any problems with it. I think that's great, and that is really what makes biology unique, and also makes it more of a long-term thing than probably any problem that a physicist is going to find. Anyway, so if you want to read more about this, Ernst Mayr, as I mentioned, had written a lot, and Simpson has written it about plants on speciation and systematics and so on. Coyne and Orr, more recently from 1980s onward, have written tremendous amount on speciation. Do read that up. Really great stuff. Even more recently, Mike Arnold has been, your teacher for this course, has been writing a lot about impact of hybridization and integration on adaptation and divergence and so on. Jim Mallett has been writing a lot about species and subspecies concepts, especially as applied to butterfly systems, but generally some of his ideas are quite widely applicable. And then there has been a lot of literature more recently about subspecies concept in systematics, what to do about it, its utility and so on. So these are various things that I have touched upon. The last thing I want to mention is we are having a very exciting symposium at the end of November, 28th to 2nd December, on genetics of adaptation. If you know any colleagues who might be interested in this, anybody who is a professional evolutionary geneticist who is working on a specific trait, knows something about genetics of it, or is a good theoretician, for example population geneticist, do let them know we are organizing this. If you just search genetics of adaptation, this will come up. We are really excited about this. We have a superb lineup of some of the best evolutionary biologists and geneticists who can integrate a lot of development that has happened in the last 20 years or so. So we are looking forward to that. And if you want more information about what we do in my lab, go to biodiversitylab.org, and under research and publications, you have all the PDF files of everything I have published so far, and under research there is pretty good description of sorts of things that you do. If you're interested, just get in touch with me. Big thanks to Janvi Joshi, Dipendranath Basu. These are two people who have participated a lot in a lot of this population to species continuum work. that we are doing, and then of course other people, Atulya and Bhavya, are more recent PhD students who are following up on a few of the mimicry themes. Collaborators I have already mentioned, and I'll stop with that. Thanks again for inviting me, and good luck with the rest of the course. I really enjoyed this course last year in NCBS, and I'm glad that that interaction with Mike has resulted into something more long term with ICER and others in India. So hopefully we'll do more and more evolutionary work here. Fortunately, unfortunately, in India there has been too much emphasis on ecology and conservation, not so much on other things. But we have tremendous opportunities, academic or otherwise, in other fields like evolution biology, and I would like to see growth in those areas. So hopefully some of you will get into this. So let me know if you want to talk about this any point, either about things that I've talked about or anything else really in biology. Okay, thanks.